Very good evening to all the NEET aspirants. This is your Prasanna ma'am from NEET World. Now stay with me for another 20 minutes to explore all the assertion, reason and statement based questions from the most challenging chapter, that is molecular basis of inheritance. Let us get started, students. So yes, the first question from assertion, reason background. Before getting into the question, let us once remind the options what you have to choose for assertion, reason, right? So option 1 is where assertion and reason will be correct and reason is explaining assertion, then you have to choose option 1. Assertion and reason are true and reason is explaining assertion, then you have to choose option 1. Option 2 you will choose when assertion and reason are true and reason is not explaining the assertion. Option 3 when assertion is true and reason is false. Option 4 when assertion is true, when assertion is false and reason is true, or both are false, you will be choosing option 4. Yes, now we will enter into the first question. Assertion wherein the genetic code is degenerate. Yes. What is degeneracy code? What is the term degenerate mean? The genetic code is degenerate, which means like one amino acid is coded by many codons. For example, if you take serine, leucine, arginine, which are these amino acids are coded by more than 5 codons. These are coded by 6 codons. If you look into glycine, valine, proline, alanine, these are the amino acids which are coded by 4 codons. Most important ma. Okay. So what is degeneracy code or degenerate for genetic codon? It means like one amino acid is coded by more than 1 codons. Reason it is explained like most amino acids are coded by more than 1 codon. Reason is also true and this is explaining the meaning of degenerate. So now you tell me what option we have to choose. Of course option 1 right. 1 right ma? Yes. And now here if you look into the second question, clear with the degenerate codon? What is it? One amino acid coded by more than 1 codons. Most of the amino acids are coded by more than 1 codon and this is explaining the assertion where he said that genetic code is degenerate. Clear with question 1? Now question 2. mRNA has some untranslated regions that are not translated. What are these untranslated regions and where are they present? If you take an mRNA strand, this structural gene will have 5 prime promoter end, this structural gene will have 5 prime promoter end and 3 prime terminator, 5 prime promoter, 3 prime terminator and in between the structural gene is there, right? So the regions which are before the promoter region and after the terminator regions are called as untranslated regions, UTR. What are these called as beta? UTR, untranslated regions. Regions before 5 prime end of the promoter and regions after 3 prime end of the terminator. And these codons will release some regulatory sequences. They will release some regulatory sequences which will bind to the operator region of the structural gene. Either they can promote the reaction or they can inhibit the reaction. They can promote the process or they can inhibit the process. The regulatory genes are produced from the untranslated regions. Regulatory genes are produced from untranslated regions. Now let us look into it properly. mRNA has some UTRs that are not translated, correct? Translation happens from promoter to terminator region only. UTRs are required for efficient translation. True. UTRs are required for efficient translation. It is because untranslated region will give some space for the promoter region so that ribosomal RNA can come and bind properly to the 5 prime cap region during the time of translation, and as they are releasing some regulatory sequences, they can also make the translation process more effectively. The role of the regulatory genes or sequences you will understand more in the gene expression. Clear beta with the second question? So here also the assertion is true and reason is also true, but in reason no way UTRs are explaining with respect why they are not translated. It is not explaining, it is just passing out a statement that UTRs are required for efficient translation. Nowhere it is explaining why UTRs are not translated. So that is why here we have to choose the option 2, where reason is not explaining the assertion. Now you can ask me a question, ma'am, how should we identify whether reason is explaining assertion or not? Just put on few questions beta, like as, if, why, how it is happening. Put on few questions after reading the statement of assertion, then you will understand whether reason is explaining it or not. Else if you are not doing that, you will be confused. If two statements are correct, you will be confused between option 1 and 2. So put few questions like how, what, why after reading assertion statement, then you can easily analyze whether reason is explaining assertion or not. Clear? Next, third question, if you see assertion, DNA fingerprinting is very well known for its application in paternity testing in case of disputes. Yes, one of the prominent applications of DNA fingerprinting is clearing the paternity issues. Second, reason. It employs the principles of DNA polymorphism. What is DNA polymorphism? Poly means what beta? Many. Morphism means structures. Yes, the small strands or segments, small DNA segments or genes of the repetitive DNA strands are of many structures, are of many different variant sequences. Based on that only DNA fingerprinting is working. Yes, true. DNA fingerprinting major application is to solve paternity issues, and it is employed because it is following the principle of DNA polymorphism. Here reason is explaining assertion, so we have to go with the option 1. Clear with these three statements ma? See, parallelly we are covering the concepts too. Stay focused ma for another 15 minutes. All the assertion, you can explore most of the assertion based and statement based questions. Here if you look into this question, DNA acts as genetic material in all the organisms? Wrong. In few organisms like retroviruses or plant disease causing viruses or organisms, the genetic material is RNA, not DNA. In this case assertion is false. It is double stranded biomolecule in most of the organisms. Yes, of course, DNA is a double stranded, be it circular or linear form of DNA, circular or linear double stranded DNA. It can be either circular or linear. It can be either what ma? Circular or linear double stranded DNA in most of the organisms. Reason is true. When assertion is false and reason is true, our option is 4. Next, fifth question, if you see beta, DNA has two chains that are anti-parallel to each other. Yes, of course, it has two chains which we will call double helical model of DNA, which are anti-parallel to each other. One runs from 5 prime to 3 prime, another will run from 3 prime to 5 prime. Okay. In one chain of DNA, at one end has free phosphate moiety at 5 prime end of the ribose sugar. Yes, ribose or deoxyribose, it is a pentose sugar, right? It is what sugar beta? Pentose sugar. So yes, it has 5 carbons, 1, 2, 3, 4 and 5. At 5th carbon, phosphate group is attached, which we will call it as 5 prime end. And at 3 prime end, there is OH group present. This gives 3 prime end of the double stranded DNA chain. Clear ma? So this is what 5 prime and phosphate group, and at 3 prime and OH group will be there. Here at 5 prime and phosphate group will be there, and at 3 prime and OH group will be there. So yes, 2 statements are correct. First statement in assertion, DNA is anti-parallel to each other, he is saying. Why is it anti-parallel? At one moiety 5 prime end is there, and at the other free 3 prime OH group is present. Here reason is explaining assertion, so of course it is option 1. Okay. Next question, if you see, adenine cannot pair with cytosine. True, adenine pairs with thymine with 2 hydrogen bonds, guanine pairs with cytosine with 3 hydrogen bonds. In reason it is like adenine and cytosine do not have complementarity between their respective hydrogen donor and hydrogen acceptor sites. Very correctly reason is explaining the assertion why adenine is not pairing with cytosine. Reason is clearly explaining the assertion. So here again the option we have to choose is 1. I have given more of reason explaining the assertion questions because you will get confused with that option only. More of those questions I have incorporated here beta, so look into it keenly. Assertion here in this question, histones are basic in nature. Yes, we know. Why they are basic in nature? These are rich in amino acids like lysine and arginine. Now you got to know how you have to choose. Here again reason is explaining assertion, we have to go with option 1. Here you see heteronuclear RNA is larger than mRNA. What is heteronuclear RNA? Heteronuclear RNA is that RNA where you will see after the end of eukaryotic transcription. After the end of eukaryotic transcription, 5 prime end and 3 prime end, which is in between, you have exons and introns, exons and introns, exons and introns, right. So this is heteronuclear RNA. When this RNA is subjected to post-transcriptional changes where 5 prime capping is done, 3 prime polyadenylation is done, and introns, which are inactive sequences, are spliced by SNRPs. Okay. After splicing introns, the length of the heteronuclear RNA will get precised and it has only exons, expressed sequences. Do remember this all the time, it is rhyming, no beta? Intron inactive, exon expressive. Okay, this is in between 5 prime and 3 prime. 5 prime which is capped with methyl guanosine, 3 prime which has polyadenylation tail. So this length is smaller than this. Fine. Messenger RNA or matured mRNA length is smaller than hnRNA, which is nothing but hnRNA is larger than mRNA. Now this concept is clear now. hnRNA has non-coding introns, true, which are not required for translation. Correct. See, in assertion he said that hnRNA is larger than mRNA, and in reason he is explaining you why it is larger than messenger RNA. So again we have to go with which option beta? Option 1. Here you see the next question. It is about polycistronic mRNA. Many a times the question is asked with respect to this point, polycistronic mRNA. You have to know what is polycistronic mRNA. Polycistronic mRNA is seen in prokaryotes or eukaryotes beta? It is seen in prokaryote or eukaryote? Tell me. It is seen in prokaryotes, wherein the mRNA sequence can be associated with many ribosomes. Okay, many ribosomes are associated with the mRNA to conduct translation. As a result, many polypeptide chains are formed. Many different protein expressions are being formed. This is called as polycistron. Many cistrons or structural genes or expressed genes are formed. Expressed protein chains are formed, that is polycistronic mRNA, where you will see only in the prokaryotes. In eukaryotes you will see what beta? Monocistronic mRNA. Keep it in your mind clearly. Polycistronic mRNA is capable of forming a number of different polypeptide chains. True. Why it is forming number of different polypeptide chains? Polycistronic mRNA has terminator codons also. Yes, it has terminator codons which will stop the process of translation. Here reason is there, but it is not explaining assertion. Here reason is not explaining assertion. Fine. If it is explaining assertion, this reason will have a statement how the polycistronic expression or how the different polypeptide chains are formed. That statement has to be explained in reason, then only we have to choose the option 1. Now as it is not explaining, it has just passed out a statement that polycistronic mRNA has terminator codons, which is a correct statement, but nowhere it is explaining assertion. So we have to choose the option 2. Clear? Next. Yeah. Here if you look into beta, assertion, in transcription, the strand with 3' to 5' polarity acts as template strand. Let us once recall the diagram where we have, right. So yes, 5' to 3' and leading strand towards the replication fork it will form 5' to 3', 3 to 5 again, and lagging strand which forms away from the replication fork. Okay. Yeah. So in transcription, the strand with 3' to 5' acts as template. Yes, 3' to 5' is acting as template. The enzyme RNA polymerase catalyzes polymerization in only one direction. So this is the diagram of replication, right? From this we will take this strand for transcription. In transcription, yes, of course, this 3' to 5' end is the template, 3' to 5' end is the template and acts as template, and this is a coding strand, right? From here again 3', 5' to 3'. The synthesis takes place always from 5' to 3'. Synthesis takes place from what direction to what direction, ma? Always, be it RNA synthesis or DNA synthesis, synthesis will always takes place, or polymerization is always taking place from 5' to 3' direction. When the synthesis is from 5' to 3' direction, template will be of 3' to 5' only. So yes, statement 1 is true. The enzyme RNA polymerase which catalyzes transcription, the polymerization is only on one direction, that is 5' to 3'. Two statements are correct, but nowhere it is explaining assertion, so we have to choose option 2. Clear? Next, heterochromatin is transcriptionally inactive. Why it is transcriptionally inactive? Because it is densely packed. Here we have to choose option 1. Clear? Next, viruses having RNA genome are shorter lifespan, mutate and evolve faster. Why the organisms or viruses which has RNA genome has shorter lifespan and they are mutating and evolving faster? It is because RNA is unstable and therefore it is mutating faster. It is because RNA is unstable. Here again reason is explaining assertion, no beta. So you have to choose the option 1. Clear? Next. Now let us look into few of the statement based questions. We will look into few of the statement based questions. Michelson and Stahl proposed a scheme of DNA replication. Wrong. DNA replication mode saying that DNA is conducting semi-conservative method of DNA replication. DNA is conducting semi-conservative method of DNA replication is said by Watson and Crick. Is said by whom? Watson and Crick. Just by looking into these two scientists and the scheme of DNA replication, don't look into that statement is correct. Don't be in a hurry when you are attempting statement based and assertion reason questions, okay. You have to look into it properly. The scheme of DNA replication is proposed by Watson and Crick, and Michelson and Stahl gave an experimental proof. What did they do beta? They gave an experimental proof. Experimental proof. By a model organism, by conducting experiments on E. coli. What is their model organism? E. coli. This you have to remember. Who proposed DNA replication, saying it as semi-conservative method of DNA replication? Watson and Crick. And who gave the experimental proof for semi-conservative method of DNA replication? That is Michelson and Stahl, who worked on E. coli. Clear? Now, statement 2. So, statement 1 is wrong. Statement 2. After the completion of replication, each DNA molecule would have a parental and one newly synthesized strand. Of course, they will have one parental and one newly synthesized strand. Just a second. Yes, they will have one parental and one newly synthesized strand. These are parental strands. Okay, and newly synthesized strand, as I said you, synthesis always takes place from 5 prime to 3 prime direction. Synthesis always takes place from 5 prime to 3 prime direction. 5 prime to 3 prime direction. Here the arrow should be over this side. 3 prime to 5 prime, right? Here the arrow should be this side. This is the parental strand and this is the daughter strand. So, for the two parental strands, daughter strand is always, or newly synthesized strand is always from 5 prime to 3 prime direction. So, you can see it has, each DNA molecule would have one parental strand and one daughter strand. This scheme was termed as semi-conservative method of DNA replication. When statement 1 is true, statement statement 1 is false, statement 2 is true, then you have to go for option 3. Okay. What are the rules for statement based questions beta? If two statements are correct, then you have to go for option 1. Okay. If first statement is correct, second statement is wrong, then you have to choose option 2. First statement is false, second is correct, then you have to choose option 3. Two statements are wrong, then you have to choose option 4. Keep it in your mind. Okay, based on that we are going to solve now. So, this is about first question. Second question, there is a definite region in E. coli DNA where the replication originates. Such regions are termed as replication fork. Wrong. You tell me children, what are those regions called as? Those regions are called as origin of replication site. Those regions are called as what beta? Origin of replication site, not replication fork. So, here also statement 1 is wrong. Statement 2, if you look into, on one DNA strand, that is template strand, 5 prime to 3 prime, the replication is continuous, while on the other, that is from 3 prime to 5 prime, it is discontinuous. For this we have to verify our NCERT diagram only. So, if you look into our NCERT diagram, yes, if you look into it properly, it is as such. Okay. So, yes, from 5 prime to 3 prime. Of course, it will be in this direction, 3 to 5. These are the parental strands. And how we have to remember, ma'am, whether it is leading or lagging? Always do remember, with respect to NCERT diagram I am saying, leading strand is towards the replication fork beta. Leading strand is towards the replication fork from 5 prime to 3 prime. When leading is towards the replication fork 5 prime to 3 prime, the template will be 3 to 5 only, no? Yes, clear? Now, lagging is against the replication fork from 5 prime to 3 prime. Synthesis of any strand is from 5 to 3 only. Okay, but do remember, leading strand or continuous strand is from 5 prime to 3 prime towards the replication fork. So, its template is from 3 to 5. So, now the statement is wrong, no? On one DNA strand, the template will be 5 to 3 and the replication is continuous, he is saying. The template is 5 to 3 means it should be discontinuous strand synthesis, not continuous strand. Okay, and other template with polarity 3 to 5 it is discontinuous, with polarity 3 to 5 it is discontinuous. So yes, this statement is also wrong. 2 statements wrong, then we have to go for option 4. Here you have to remember one thing, template strand from 3 prime to 5 prime, synthesis will be in a continuous way from 5 prime to 3 prime direction. Okay, discontinuous strand synthesis is away from the replication fork from 5 to 3, the template is 3 prime to 5 prime. Clear? Next. Yeah, this is a statement if you observe, the DNA polymerase on their own cannot initiate the process of replication. True. What is initiating the process of replication? The enzyme primase. The enzyme primase will synthesize RNA primer. RNA primer. RNA primer we have discussed, no beta. RNA primer is the starter. It is the starter of the process DNA replication. Then DNA polymerase will take the initiative and it will continue the process of DNA replication. So yes, this statement is true. Saying that DNA polymerase cannot initiate process of replication is true. Average rate of polymerization of DNA polymerase has to be approximately 200 base pairs per second, he is saying. No, it is not 200 base pairs per second, it is 2000 base pairs per second. See how important your NCERT statements are, it is directly asked from this. So yes, this is wrong. When statement 1 is 2 and 2 is false, then you have to choose what option beta? Option 2. Next, both the strands of DNA are not copied during transcription, correct? Only one strand is copied, right? Not both the strands. In transcription only a segment of DNA and only one of the strand is copied. Let us take one segment, that is 3 prime to 5 prime, that is template strand of DNA replication, 3 prime to 5 prime, and of course leading strand which is continuous strand will be from 5 prime to 3 prime. Okay. Now during the time of transcription only one RNA strand is synthesized, right? One RNA strand is synthesized that is complementary to 3 prime to 5 prime. That is complement, this is from 5 prime to 3 prime. Just a second. Template strand 3 prime to 5 prime. Then you tell me what is the leading strand? Leading strand will be from 5 prime to 3 prime. Now new mRNA synthesis as I said you will always happen from 5 prime to 3 prime. In that case it will be complementary to this strand, right? So yes, synthesis will take place from 5 prime to 3 prime. This is mRNA. This is complementary to template strand. Template strand means this strand. This is the template strand. Template strand is also called as sense strand and non-coding strand. Okay, as the sequence of mRNA is same as that of the coding strand, is same as that of this strand, this is called as coding strand. Except in the place of thymine, here it will be uracil, rest everything will be same. So this is also called as, it is same as that of coding strand. Coding strand is also called as anti-sense strand. Okay, clear? So yes, if you look into two statements, both the strands of DNA are not copied, correct? Both the strands are not copied, and in transcription only a segment of DNA is taken and only one of the strands is copied into RNA. One of the strands, what is that? Template strand, and it is same as that of coding strand. Do remember that. Yes, both statements are true, then we have to go for the option 1. Next, this strand that has the polarity 3 prime to 5 prime acts as template, correct, and is also referred to as coding strand. 3' to 5' polarity, this is called as template strand, not coding strand. So this is wrong statement. Next, the promoter is said to be located towards 5' upstream of the structural gene. This is the structural gene beta, and with respect to the coding strand or structural gene, promoter region is present towards the 5' end, okay, and terminator is towards the 3' end of coding strand or the structural gene. Clear? So yes, this is true when statement 1 is incorrect and 2 is correct. Here we have to choose the option 3. Clear? Next question. Few more questions beta, be there. Yes, here RNA polymerase 2 transcribes precursor of mRNA, that is the heterogeneous nuclear RNA. True. Splicing is the process where exons are removed and introns are joined. If you remember, just now I said exons means expressed sequences. Exons means what ma? Expressed sequences. Introns means what? Inactive sequences. Exons means expressed sequence, introns means inactive sequence. So yes, introns are spliced, exons are not spliced. So this is wrong. When statement 1 is true and 2 is false, you have to choose what option? Option 2. Okay. Next, AUG has dual functions. It codes for methionine, correct, and it also acts as initiator codon. True. Statement 2, if you take, stop codons or terminator codons are UAA, UAG and UGA. 2 statements are correct, so you will go for option 1. Next question. The replication occurs within a small opening of DNA helix referred to as replication fork. True. The DNA dependent RNA polymerase catalyzes polymerization only in one direction, that is 5' to 3'. I already said you polymerization happens in 5' to 3' only. True. But now I would like to ask you, DNA dependent RNA polymerase he is saying. DNA dependent RNA polymerase means it is conducting replication or transcription. Of course it is transcription. Because in transcription RNA strand has to be synthesized, so RNA polymerase is present, and this RNA polymerase is dependent on DNA strand, so that is why we are calling it as DNA dependent RNA polymerase. So yes, this is true, this is also true, then we will go for option 1. Clear ma? Yes. Next question. Both the strands of DNA are not copied during transcription. He is saying what? Both the strands of DNA are not copied during transcription. Correct. Just now we have discussed, no, only one strand is copied. What is that? Template strand. In transcription, only a segment of DNA, and only one of the strands is copied. True. I think the question is repeated. Yes, these questions are repeated. You look into this question beta. Yeah. A gene is defined as functional unit of inheritance. Correct. Gene is defined as functional unit of inheritance. The DNA sequence coding for tRNA, rRNA molecule cannot be defined as a gene, which is wrong. Gene is nothing but any expressed part of the DNA or any functional segment of the DNA is called as gene. So yes, gene which is coding for tRNA and rRNA, it is also called as gene. It is coding or doing an activity, no, a function, right? So it is a functional part of a DNA, so it is called as gene. Okay. So this statement is wrong. Statement 1 is true and 2 is false, then you have to choose the option 2. Clear? Next. Yeah. The cellular factory responsible for synthesizing protein is ribosome. Correct. The ribosome consists of structural RNAs. Yes. Ribosome consists of 2 subunits and many structural RNAs. About 800 different proteins. He is saying wrong. Approximately it can have around 80 different proteins. So statement 1 is true, 2 is false. You have to choose which option beta? Option 2. Look into this question. Z gene codes for beta galactosidase, that is primarily responsible for hydrolysis of disaccharide lactose into monomeric units galactose and glucose, he is saying. What is this Z gene? Where do you see it? If you take Lacoperon model, promoter, operator, later you will have structural genes Z, Y, A, and then terminator. Okay, promoter, operator, structural genes and terminator from 5 prime to 3 prime. This is the mRNA gene, right? mRNA sequence, right? So structural gene, right? So here yes, Z structural gene is coding for the enzyme beta galactosidase. Y is coding for permease. A is coding for transacetylase. Z, which is coding for beta galactosidase, its function is to cleave the disaccharides into galactose and glucose. Yes, statement 1 is true. Lactose is substrate for enzyme and beta galactosidase. Okay, it's the substrate for enzyme and beta galactosidase, and it regulates switching on and off. In the presence of lactose, it is switching on the process, right? The presence of lactose, it is switching on the process. In the absence of lactose, the process is off, because at the operator region, repressor will bind and it will block the process of transcription. So yes, this is true. When both the statements are correct, you have to go for the option 1. Clear? Next. So here lactose is responsible, right? Whenever lactose is present, lactose is binding with repressor. So nothing is there to block the process, transcription is continuing. Whenever lactose is absent, repressor going and binding to the operator region, it is blocking the process and transcription is not happening. Next, the commonly used host were bacteria and yeast, and the vectors were called as bacterial artificial chromosome, yeast artificial chromosome. Of course, they are most commonly used vectors, right? Yes. Statement 2 he is saying like DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repeat DNA, because in the sequence a small, just a second, the question is not clearly available. Yes, he is saying like in statement 2, DNA fingerprinting involves identifying different sequences of the specific regions in the repetitive DNA. We have already discussed, there are small repetitive DNAs, and in that each sequence is variant, okay, variation of the DNA sequences and genes, that is called as DNA polymorphism. Based on this, DNA fingerprinting is done. Yes, this is true statement, and the most commonly used vectors are bacterial artificial chromosomes and yeast artificial chromosomes. This is also correct. When two statements are true, we have to go with the option 1. Yeah, last part of the statement based question, stay focused. Radioactive phases were not allowed to attach E. coli bacteria. Wrong. Radioactive phases can attach the E. coli bacteria. It can attach to E. coli bacteria. Both RNA and DNA are not able to mutate. Wrong. Only DNA will not mutate, RNA can mutate. When two statements are wrong, you have to go for the option 4. Next question. The promoter and terminator flank the structural gene is a transcriptional unit. True. The coding sequences are expressed sequences and defined as introns. Wrong. Many at times, do remember students, the questions are asked on introns and exons. You have to remember it and fix it in your mind that introns are inactive sequences, exons are expressed sequences. When statement 1 is true and 2 is false, then you have to choose the option 2. Okay. Next. The RNA polymerase is only capable of catalyzing the process of initiation. Wrong. If you remember the 3 steps in transcription, that is initiation, elongation and termination. At the time of initiation, RNA polymerase is binding with sigma factor forming a holoenzyme, right? During the process of termination, RNA polymerase is binding with rho factor, and here the termination occurs. And elongation process is catalyzed by RNA polymerase only. So here RNA polymerase is not just for initiation, it is wrong. The accessibility of the promoter region of eukaryotic DNA is in many cases regulated by the interaction of proteins with the sequences called as operators, he is saying. Wrong. Okay, it is many times regulated by the interaction of proteins. Once again, let me read the statement. The accessibility of promoter region of eukaryotic DNA is regulated by the interaction of proteins with the operator. Whenever such questions comes, first draw the diagram where you have learnt in gene expression. First promoter comes, then the operator comes, then structural genes you will see, Lacoperon model, then it is terminator. First RNA polymerase has to come and bind with the promoter region, right? RNA polymerase has to come and bind with the promoter region. Then the process is started, and to the operator region, if repressor is binding, it will stop the process. Okay, it will stop the reaction, it will stop the process of transcription. So now you read the statement again. The accessibility of promoter region is in many cases regulated by interaction of sequences with operator. No, the accessibility of operator is based on the promoter region, because promoter is starting the reaction and promoter is prior to the operator. Okay, promoter is prior to the operator. Now what is starting the process? RNA polymerase binding with the promoter, it will start the process, not the operator region. Okay, so yes, this statement is wrong. So when both the statements are wrong, you have to go for the option 4. Yes, that's it beta. With this we have, that's it children, we have, with this we have covered up or explored most of the concepts covering statement based questions and assertion reason questions. If you are having any doubts regarding this, please place all your doubts in the comment section. I will be answering all your doubts. And before ending the session, I would like to announce a very important thing, or which is useful for all the enthusiastic students who want to attempt NEET 2025. Here in NEET World, that is your step to medicine, NEET World, which is located in Narayanguda, we are starting a crash course which begins from March 24th. Okay, and it is 40 days crash course, available both online and offline. Online, how is it available ma'am? Where we need to access? Just go to Play Store and install the app Med World. Install the app what ma? Med World. Med Space World. After installing this app, you can get into the crash courses, and you just call to that number which is given below. They will let you know about the further details. And the most important thing which I would like to say you is 10 mock tests are for free beta. 10 mock tests with the paper explanations are free. Okay, 10 mock tests with paper explanations are free. And you will have part tests. How many part tests? 5 part tests and daily practice tests based on the chapters. Okay, based on the chapters covered, you will be having daily practice tests, and each week you will be having part tests, and on whole in this crash course program 10 mock tests will be available. And one more exciting news for you is this 10 mock tests are for free. Whoever are enrolling for the crash course or not enrolling, this 10 mock tests will be free if you just install the MedWorld app, and you see the 10 mock tests will be for free. They will be accessed based on the schedule which is given. The video for the video on how to access the mock test, on which date each mock test is released in the app, will be dealt very soon. Okay, so stay tuned. Thank you.